This assay may help to clarify the role of tryptophan residues in proteins when performing ANS binding studies. The ANS fluorescence increases upon binding to specific structural sites in proteins. Sometimes, ANS binding site locates close to a tryptophan residue with a distance that suggests the existence of thread between the tryptophan and ANS. NBS mediates chemical modification of tryptophan residues in proteins, quenching its fluorescence. Therefore, this assay allows determining whether tryptophan residues contribute to the increase of ANS fluorescence intensity by thread. In this work, we use an engineered recombinant nucleotide binding domain from Ca2, containing mutations aiming to locate a tryptophan close to the nucleotide binding site where ANS also binds. This assay is rapid and easy to perform, hence its main advantage. Determination in silico of the ANS and Ca2 end-domain interaction. Generate a three-dimensional structure of the protein by molecular modeling using the preferred protein modeling software. Identify the amino acid residues that form the nucleotide binding site using the preferred molecular structural software and determine the presence of arginine and lysine residues. These are required for ANS binding and to increase the fluorescence intensity. Because of the presence of arginine and lysine residues in the nucleotide binding site of Ca2, we hypothesize the binding